My name's Katie and I'm a research technician here with the ex-situ conservation program at Kings Park Science.
In the previous video, I covered how to harvest shoot tips from our tissue culture collection.
Today I'll be preparing shoot tips for cryostorage. Let's get into it. This is a day in the life of a Kings Park scientist. Our shoot tips have been sitting in the temperature-controlled growth chamber for 2 days on desiccation media, designed to reduce their water content, lowering the risk of ice crystal formation. I'll need some featherweight forceps for a gentle grip on the extra tiny shoot tips. Here I am gathering some solutions, which are a mixture of cryoprotectants and sugar, that can help to remove the moisture in the plant cell in preparation for rapid freezing. Rapid freezing, or what we call vitrification, is a process used to protect living plant tissue that is entering cryostorage. We use different cryoprotectants during the process to prevent ice crystals from forming inside the plant cells. How exactly? Let me explain with a mini animation.
This is a happy, healthy plant cell at room temperature. Here the water molecules are free-flowing. As the temperature begins to drop, water molecules turn into ice crystals and continue to accumulate within the cell until the sharp edges puncture through the cell wall, causing rupturing and eventually death. To prevent killing the cell, we soak the plant material in cryoprotectants at low temperatures and then plunge them into liquid nitrogen at -196°C. This results in water becoming glass-like rather than sharp crystalline ice. The cell is now frozen in time, but alive. Tools are sterilised and ready to use. I feel like I've forgotten something important. What have I forgotten? Timer! Timing is very important in preparing plant tissue for cryostorage because most of the chemicals we use are to a degree toxic to the plant. We need to carefully control the concentration and contact time to make sure we are maximising the benefits of these chemicals without damaging the shoot tips during rapid freezing. Our tools are ready, our timers are set. Let's clean my hands and get started. We need to transfer our 2-day-old shoot tips into their first bath with loading solution and soak them for 20 minutes to dehydrate the cells. While we wait, we can grab some ice to prepare the second bath. The cryoprotectant concentrations are high in this solution, so it is more viscous or thicker, as you can see. I'll sit this solution in an ice tray to keep them cold. The lower temperature will slow down cell activity, reducing the rate of toxicity. I'll scoop the shoot tips out gently with the featherweight forceps from the loading solution bath and transfer them into the cold cryoprotectant soak for another 20 minutes. While they are sitting in the iced cryoprotectant bath, we can prepare the liquid nitrogen. Cryo gloves on, let's go grab some. These are special gloves, not your regular oven mitts. They'll protect my fingers from the -196°C temperature. I'll transfer the liquid nitrogen into a styrofoam box to pre-chill the cryovials inside. A cryovial is a mini tube that is built to withstand the ultra cold inside the dewar. I can maximise storage space with these vials and organise them nicely into the dewar within storage boxes. The dewar works like a giant thermos. It has a vacuum between the walls for extreme thermal insulation. We have a couple of them in the lab and we use them to store our tissue culture collection and our orchid collection. That's our second bath done. I can now prepare the foil strips. The foil acts like a miniature tray for the shoot tips to sit on while protected with a droplet of cryoprotectant, easier and much more efficient to handle. I use the featherweight forceps to gently scoop the shoot tips out and move them from the bath onto the foil. I need to make sure each tip is wrapped with a little droplet of cryoprotectant and I'll put about 10 shoot tips per piece of foil. We're finally ready for the exciting part. Using long forceps, I grab the foil and plunge it right into the liquid nitrogen, while unscrewing the cryovial cap and quickly but carefully sliding the foil into the vial and then putting the cap back on. All the vials are labelled prior to freezing with the species name, accession and date of storage. The vials are then packaged into labelled cryo boxes with a location ID. Label, label, label. Very important. This will ensure anyone that's working with our collection decades later will be able to identify the specimens stored today. I will put all the vials into their designated location within the cryo box. The lid will go on and then we're ready to move into the dewar. The cryo boxes are slotted into the rack and locked into place with a pin. And then into the dewar they go. This is how we use cryostorage to conserve our native flora at Kings Park Science. Stay tuned for part 3 to see how we take shoot tips harvested decades ago and use them to grow plants today. See you in 20 years!